All right. Doug, did you see a notification that the meeting's being recorded? Okay, excellent. So we've got that going. So the last thing I want to cover before I jump to the agenda are the workshop goals. So welcome everyone. The goals of this workshop are to learn from one another's experiences and network with your peers. The expectations of these meetings is that everyone has the responsibility to contribute. We'd like you to be an active listener and speak honestly. We want you to share information in ways that can help others be successful. Be respectful and open to new ideas and new ways of doing things, and everyone participates and no one dominates. And of course, even though we're just starting our first series of workshops here this morning, I want to promote our next workshop. So we've got one on the 4th, 13th, 21st, and then a couple in-person workshops in April that we'd love you all to attend. You have all this information in your emails. So I am recording the workshop so that people can access this information after this workshop. So we've got 8 speakers here this morning. So we'll go through and have them present and leave a little discussion after each presentation to talk about these different topics and new approaches to doing aquatic invasive species prevention and management work. And so I'm not going to read everyone off, but I will introduce everyone as they come on. So first we have Amy Kinsley with the Minnesota AIS Research Center. So Amy, if you want to start sharing your slide, you are our first presenter.
Okay, so I've shared my screen.
Yep.
You should see it with no speaker notes, correct?
Yep, we're seeing the presenter mode. Yeah, presentation.
Perfect.
Yep.
Perfect. Okay, so first of all, I want to thank you all for joining this morning and for Tina and Doug for the opportunity to present. I feel exceptionally privileged to be the first speaker of the morning because I know you all, well, if you've had enough coffee, hopefully you're all fresh and excited about the workshop and about hearing some of this material. So again, thank you for being here and taking time out of your busy day to hear my work. And so I've presented this material in the past, mainly focusing on the methods and then creating a vision towards the future for what that might look like as we integrate it into some of the tools that MACERC has developed specifically that I've been involved with. And so my goal for today is to highlight the tool component, so the tool development, and I want to interest you in learning more about it and hopefully in joining us in the future for a workshop or a one-on-one consultation. And so yep, I'm Amy Kinsley. I am a professor in the veterinary school. As Tina said, I'm also affiliated with MACERC, or the Aquatic Invasive Species Research Center, where I have been a research fellow and have been working on tools and like this for the past 5 or so years now. And today I'm going to share some updates to the AIS Explorer dashboard. And so for those of you who are not familiar with the dashboard at all, it is a free interactive dashboard that was designed with the goal to support AIS managers with decision-making around AIS prevention and management. And the goal was really to make data-driven approaches more accessible. And it's interactive, which means that it can be tailored to your interests. So if you are looking at a specific species or a specific county, you can adjust some of the capabilities within the app to get outputs that are relevant to your questions. And it also automatically updates to account for changes in lake infestation status, which is great because as somebody who develops models or algorithms that provide support, those things become outdated quite soon if it doesn't account for those aspects, right? And so it's available at www.aisexplorer.org. You can also reach it through www.aisexplorer.umn.edu, and I have those below on the slide. The dashboard was made public around 5 years ago, and this has been an initiative involving partnerships with researchers and managers The researchers have come from a variety of disciplines and institutions, and without feedback from folks who are, involved in that decision-making aspect, it wouldn't have gotten to where it is today. And so I just want to acknowledge how important that component is. So when you click on the start button on that homepage I was showing, the first thing that will pop up is this introduction risk for surveillance tab. And that won't really be the focus of what I'm talking about today, but this is the experience that you would have. It shows Aiken County, which is just because it's the top in the alphabetical list, starts with A, it's the first thing that would pop up. That is the default. And what it's showing is the results of a risk model that estimates the likelihood that a water body becomes invaded with either zebra mussels or starry stonewort, and you can make those selections within the next 5 years. And so Nick Phelps, he'll present after me. We'll chat a bit more about this and other updates. But I want to focus your attention on the tab that we call the prioritization for watercraft inspections. And so I've highlighted that here in red. If you click on the top, it will navigate you to that page. You will navigate to that page. And we have a couple of updates within this tab. And so it's meant to support mainly county managers in prioritizing lakes for watercraft inspectors based on our estimates of boater movements. And so on the panel on the left, you'll see a variety of options. You can tailor it to your county of interest, and you can select different risk species. So for this approach, we consider zebra mussels, starry stonewort, Eurasian water milfoil, and spiny water flea. And you can adjust the tab to give you outputs that might be more reflective of management goals. So in some ways, one might think like, well, I would like to be able to place out inspectors so that I'm inspecting all of the potentially infested boats along the landscape, or that are moving in and out of water bodies in my county. But of course, that may be resource intensive, depending upon your county. And so we allow you to navigate, and adjust that percentage for what your maybe interests are. And some of this would be a curiosity, maybe for information, but some of this also can be helpful in communication. So if you're printing off these and sharing this with folks, maybe you just want to focus the attention to a specific point. And I think that was a helpful element of that. But so within here, you get an output that looks like this. So you have a table here on the left, and then you have a map on the right. And here we're showing Hennepin County. I just selected Hennepin County. It is the county that I live in. But what we're saying here is that if you want to inspect 100% of the risky boats in this county, you would need to place inspectors at 126 lakes. And so this isn't access points. These are lakes, and this is the level of granularity at which our models are functioning at right now. But that also gives you a map then where you can see where those locations are. Almost every lake, right? That's highlighting here in the color. And so, so it's telling us, that 126 lakes. You have the capability then to download a CSV file, or you can get the image, the map image as a PNG. And so those are things where if you don't want to maintain this live on the website to share it, you can save it in that form and revisit it. But if you toggle over to the chart, this gives you the number of inspection stations that have been placed with the percentage of boats that have been inspected. We're kind of functioning here off this definition of risky boats, which I'll get into in our next slide. But this can also help understand, if I add more inspectors at some point, I'm not really gaining much in terms of number of risky boats. And at some point that payoff won't necessarily be worth it. And these tools have been built using boater networks. And so these networks have been generated using watercraft inspection data from 2014 to 2017. And that includes data, of course, from inspectors that have been on the ground. But we also know that boats are moving between water bodies that we aren't capturing information about. And so we've been able to overcome some of the biases in that by generating some predictive models. And so this work has been published. I have the Cal 2021 paper here, and it can give you a bit more information about that, or give you access to that paper when he presents during his presentation. But essentially, we have information about annual boat movements from over 9,000 lakes across the state. And so these surveys that we had used from the DNR had over 1,600,000 survey responses, so robust amount of data. And as I said, we were able to overcome some of the biases in the data collection process by being able to use machine learning algorithms, which have become quite common in a lot of our day-to-day life, to estimate boat movement. As I mentioned before, the app automatically updates. So as a new water body is listed as infested, the model outputs automatically account for that. And so there is a last updated timestamp on the app, so you know how recent those updates are. And then, as I mentioned before, I might use the term risky boat when I talk about some of the work. And when I say risky boat, I mean it's boats that move from infested to uninfested lakes. And so that means we've taken watercraft inspection data, we've modeled that up to account for those unobserved movements that are occurring. But really, when we get down to inspections and the way that we're thinking about it within the model, or the tools that we've developed, we're really honing in on those boats that we're considering risky, or those that are moving from those infested lakes to the uninfested lakes, and characterizing things based on that. So keep that in mind. As I said, I won't dive deeply into the methods of the algorithms that are supporting the outputs that we're seeing today within the model, but I'm showing here a couple of papers that have been published that go into much more detail. And both of these papers that I'm showing here are available at no cost. So you should be able to maybe search up the title or even my name and download it and peruse that. If you want to ask questions or gain any more insight on those, I'm always happy to go through that with folks. So that is probably one of my favorite parts of my job, is communicating with folks who use the tools that I develop, or just interested in, it has sparked something within them to ask further questions. So don't hesitate to reach out. You can also check out NASERC's YouTube channel for previous talks on the topic, where there's a bit more of the like math behind what's happening that I'm showing today. But for the sake of time, I won't get into that. And so I'll just guide you towards that. And that is available there on the bottom right-hand side of the slide. I'm gonna go through a bit of the what we're doing intuitively, just so you have an understanding of how we got here. It's not really the math behind it, but it's building some intuition around what we're doing here. Essentially, we're saying we have these boats that are moving from lake to lake. And as I mentioned, we're really interested in those that are moving from these infested to uninfested water bodies. And so I've highlighted those in red. And I have here like all these counts of boats. And this is all kind of a toy network, or just an example. And what happens is we rank these circles that we're showing here, these water bodies, based on how many boats are coming and going, those risky boats that are coming and going, and kind of rank those that are the most receiving or sending out the highest number of boats. And so we rank those as the #1 water body, meaning that if you place an inspector there, in theory, right, you would be inspecting the greatest number of risky boats that are moving within the county. And then assuming that that has been inspected and there's 100% coverage and 100% effective at it, right, then we come up with the second ranked, and then the third ranked, and the fourth ranked. So it accounts for that in those inspections. And so that's what shows up here in this table. That is how that is generated. And I just wanted to build that intuition because I think it plays into what are the new components of this, which is really what I want to share today and to kind of pique your interest around. And so within here, the new parts, if you click on the prioritization for watercraft inspections and you've been there before, you maybe have noticed these little bits weren't there and now they are. And so there's a new sub-tab, is what I'm calling them, that is called collaborations or paired planning. And the idea there is that I've investigated, well, this came up in questions and conversations that we have had around, are there ways we could be more efficient with the placement of inspectors or more effective by communicating with other counties where boats are moving in and out of counties from those infested to uninfested water bodies. And so we generated an algorithm that was able to identify what those movements were in terms of counties. So instead of looking at it from a water body, now we're focusing in on those movements within counties. And here I'm showing the two networks that were revealed from our analysis, looking at that high level of risky boat movement. And so here we see Ramsey, Washington, and Anoka County. These lines just show where those movements are coming and going from and how those connections were formed. And then in this kind of larger network, we see Cass, Crowing, Hennepin, Wright, Stearns, and Carver. And again, you start to see kind of this web of contact and complexity, and maybe not totally transparent without revealing explicitly what those lakes are, where the movement is, but kind of identifying this at a larger scale of what those are. Because within the tool, we do allow for that. So I'm showing the same network, slightly different structure. Here, I'm not showing all of the connections. I'm just showing those where the highest number of movement is occurring. But these colors for these counties also correspond to the lakes that are over here in this right panel. And so you can start to see a bit more of how those movements are occurring based on our network models. And you know, that probably confirms what you've already known, or what you maybe already have some understanding around. But I think what's interesting here is it can be a bit more explicit to open up conversations about, oh, look, like there are these strong connections between Leech Lake and Minnetonka. And then within Minnetonka, there's also a lot of these other connections that are involving lakes, right, from Carver County, like Minnewaska, and Riley Lake, right? And so it kind of builds out this web in some ways, because this is only showing the top 50 lakes, or the lakes that have like the highest number of connections. And so it's kind of opening up the conversation around these cross-county movements that are occurring, and ways that communication around this maybe could be more effective or fruitful. And so what I'm showing here is an output, as I showed you before in the table, only instead of one county's output, I'm showing the network of counties. So this is assuming that all of those counties that were in that larger network, there's no boundaries there. Like, we're just ranking them as if movements are flowing freely, and we don't have any priorities based on our specific county interest. And if we were, though, to think about, well, if I were to partner with somebody and see the impact of sharing information or resources, what might I do? We have developed a tool called paired planning. And so this gets quantitatively complex. And so at this point, we're only able to do it at the paired stage. But essentially what this allows you to do is go in and select lakes to say, yep, I'm going to put inspectors at these locations. What does that mean then for my neighboring lake, or neighboring and maybe not contiguously neighboring, but maybe we share large numbers of boats moving between us? What does that mean for their planning process? And so here we have the first county. This is where you're going to place the inspectors, and you're going to assess the impact. And here, if you click select lakes for inspections, you can go in and choose those things. Here I've chosen some of those that were already shown to be quite popular lakes, where you would get a significant benefit out of placing inspectors anyways. And you can see in the next slide that what that means in terms of your priority list. And so this wasn't maybe a hugely remarkable change, but I am just highlighting here that what that did was bump down Bay Lake and Edward Lake in terms of priority for Crow Wing County. But you could imagine that was just kind of a simplified analysis of what could be done, and also maybe just a way to initiate conversation around things like this and help foster some collaboration around watercraft inspection planning. And I see I'm running short on time, but I just wanted to whet your appetite. So if you were interested in hearing any more about this, I want to encourage you to reach out. This is available for all counties, whether you're part of a collaboration network or not. You won't be able to visualize that network of top 50 lakes, but you can go in and see the impact of inspection planning on one versus the other county for all the counties in Minnesota. Next steps, we want to explore information sharing, research sharing, update the networks. So we know that we realize the networks were built from 2014 to 17. We have data from 2018 to 2023 now, and we have a graduate student who is working on that. These workshops I want you to participate in, we're hoping to launch early fall. So if you are all interested, I encourage you to reach out to Maddie Hayden at MACERC, and I have her email address here. And again, if you ever want to do a one-on-one consultation, help me refine the way I think about these things, develop new tools for the future, gain a stronger understanding of the tools that I presented today, please do not hesitate to reach out. I also want to acknowledge that this work was not done in isolation. As I said, there's an interdisciplinary team who's been contributing this, and I'm only just showing the fraction of the portion that I highlighted today. And this was built on feedback and ideas that were generated from conversations from a wide range of folks, including county managers and folks from the DNR and other collaborators from the University of Minnesota. And I just want to highlight that their contributions were important. And with that, I would like to conclude my presentation.
Thanks, Amy. I know I saw one hand up, so why don't we take one question from Russ before we move on to our next speaker?
Any idea how soon you'd have the updated inspection data incorporated into it? And then I was also wondering if the actual infested waters was up to date, or if that's also from 2017.
So the infested waters has been updated. If you're in AIS Explorer, it should have a timestamp on there. I believe when I was looking at it earlier, the most recent update was from October 15th. And so that's pulling from the DNR's infested waters list. And so if everything in our pipeline is working correctly, then that should be the last update from the DNR's infested waters list. In terms of the new information about the watercraft inspection data, I would anticipate that that would be available sometime this spring. That is the goal. We want to create protocols for when we need to update again. So we're being really thoughtful about this process of kind of the sustainability of these networks and what that means when we continue to update them, and also investigate or ask questions about what's changing across that time period. So we're hoping to reveal some of that as well as we move along, but we'll also be in communication. So if we change those within AIS Explorer, we will make sure that folks who are users are aware of those changes. It wouldn't be just the network sneak in and everything changes and you aren't aware of that. So yeah, sometime this spring, and we'll make sure that we create a lot of awareness around that, because I know folks are really eager to hear about that.
Thank you, Amy. I'm going to kick it on over to Nick, and he's got his presentation up and shared, so that's great. Glad we figured out that technical issue there, Nick. Take it away.
All right. Yeah, apologies about the presentation. It was a privacy setting. I didn't realize I had turned on. So thanks for having me, and good morning, everybody. I'm going to build on Amy's talk here and share another new aspect to the AIS Explorer. And this one's looking at the cost benefit of intervention, specifically the prevention activities that many of you are doing across the state. All right, so first, the acknowledgement, as Amy pointed out, this has been a huge project over many years, and there's a lot of really smart people. Those in red are the students that have helped along the way. And I also want to thank, like Amy did, many of you who have provided comments and insights and guidance as we've aimed to develop these online tools. Our goal has been to take big data sets, complicated systems, distill them down into user-friendly applications that folks like you can use. So that's been our aim, and we've progressed through a series of apps to do that. These are some of the data sources and models that we've talked about in the past. Amy mentioned a few of them. I don't have a time to go into any of this, but like Amy pointed out, a lot of this has been presented. There's papers published. I'm happy to share any of those resources if you're curious to learn more. But where I want to focus here is an aspect that has been missing, and we've heard this loud and clear from many of you, that when we intervene within these networks of boater movement, we have the opportunity to reduce risk on the landscape. Now, we know this to be conceptually true, but we haven't had a way of incorporating that into the AIS Explorer until now. So this is provides some usability and functionality to these networks to really manipulate them. So I'm going to walk you through sort of a diagram here of how this works, and then I'll show you the application and what's available online right now. So the way you might sort of view the current risk models that are on the AIS Explorer, the surveillance tab and then the watercraft inspection tab, we're assuming status quo management across the landscape. That means that any boat that's leaving any lake, regardless of what intervention's happening there, is all equal. So if 50 boats are leaving an infested lake, all 50 of those are going somewhere else and still potentially risky. That's how it is now. But what we know to be true is that as we intervene, we can have an opportunity to reduce risk in a lot of different ways. So let's, here, just at this high level version with interventions, boats that are leaving an infested lake have some sort of intervention, inspections, decon, education, and then that same or different type of intervention might happen at a receiving lake. And so conceptually, we can think this through as 50 boats leave, and some fewer number of boats, of risky boats, are entering another lake. Now, this is how it's working in the real world. And we've worked over the last couple of years to try to put real numbers and estimate how well these interventions work, what the costs are of those various interventions, and really quantify the risk reduction across Minnesota. So I'm going to give you one more step of complexity here before we go into the actual tool. Now imagine we have these 50 risky boats, and they're all leaving an infested lake. So again, we define risky boats as any boat that's leaving an infested lake. And we do watercraft inspections at that site. So those 50 boats have an opportunity to be inspected. We spent the last 2 years trying to quantify how effective these different interventions could be and what those costs are for each of those interventions. So in this case, as it relates to the watercraft inspections, we've estimated through interviewing county managers that the average cost for a watercraft inspection is about $20 across the state. That could be more or less depending on how many boats are coming through there, but for now, just for this example, think of it as $20. And let's say that inspector that's there inspects 20% of those 50 boats, because they're not there every day or all day. And so some boats are getting through. And in this case, they're inspecting 10 of them. And the effectiveness, we've estimated through on-site trials at boat ramps, a watercraft inspector is roughly 80% effective at removing invasive species from the boat. Now, at this step in the pipeline, we've spent about $200 to get those 10 boats inspected, and we've reduced the risk, if you multiply effort by effectiveness, by 16%. So now we have approximately 42 boats that are moving around the state from that infested lake, first step of risk reduction. Now, let's say these boats go into a second lake and they're subjected to educational signage or pamphlets or a billboard as they come into the access. We've estimated that the cost to sort of provide that educational opportunity to boaters is about 40 cents per sort of exposure. The effort, let's say, is 70% because it's a lot easier to do, and maybe 70% of those 50 boats are seeing that billboard as they drive into the access site. And we know through our on-site inspections of testing volunteer boaters at boat ramps that they can remove about 50% of the invasive species from their boats. So just moving really quickly through this gives us a cost, let's say in this hypothetical scenario, of $14 to do that education, to get those 50 boaters of an opportunity to be exposed, and a risk reduction of 35%. What you get in the end is 24 boats going into this final lake. Now, these steps in this process, this is just an example for just a very simplified version to explain it to you. But imagine doing this for every lake that every lake is connected to in Minnesota. And that's where the power of these massive algorithms comes in, and these huge data sets, and brilliant folks like Amy and Alex who have managed to figure out how to use these numbers and put them online for managers to play around with. So again, this is just the conceptual version. What I'm going to show you now is what it looks like in practice with all of these connections and all of these data going on behind the scenes. So here again is the AIS Explorer. Amy talked about this. We'll just jump right into the Intervention Impact tab. Now, if you haven't been on the website for a few months, this is new. This has been added, so it's up there on the top. I've got it circled in red. And you're going to walk through a series of 5 steps to sort of choose your own management adventure. Create a scenario that you think is appropriate for any given set of lakes in the state. So this is ideally designed for county-based managers, but everybody else can use it. It's free online, play around with it all you want. Maybe not all at once right now because it'll probably crash the website, but if it is slow, come back later and have a look. This is hard to show you in a presentation like this. Much better to look at it on a website. So I'm going to show you some still screenshots, but imagine that there's other things happening here up and down if you were to scroll on these pages. So on this the first step in this process is to choose your lakes. You can narrow this down by county and select any given lake in that county or all of them that you want to apply some version of intervention to. Now, this is how managers would sort of the first step. Where do I want to intervene? And you can focus on infested lakes as sort of a containment strategy, uninfested lakes to protect them, some hybrid of the two. This is totally up to you. Here in this example, I've picked Crow Wing County. We've picked 5 lakes to apply some more stringent interventions, but we're going to allow this model to do education at every lake in the county. The next step is defining the effort. Now, when we think about sort of the effort that a watercraft inspector or a decon or education, this got a little bit tricky for us, and I'll challenge you to think about effort in the context of this tool as the number of boats that you're going to have an impact on. So if there's 100 boats going to a lake and you think you can actually talk to 10 of them, that's 10% effort. If that makes sense. So in this tab here, we have seeded default values for all of this based on data that we've collected from counties and the DNR and interviews with folks to be somewhat reasonable. But you can go in and modify any individual lake or all lakes at once in this tool. So really, this is a choose your own scenario experience. The next step is to look at effectiveness. Now, I mentioned earlier, we've quantified this over the last few years on how effective each one of these interventions are. And the ones we have built into the tool right now are watercraft inspections, hot water decontamination, and boater education. Now, these are seeded with empirical data to suggest that these might be averages statewide, but we know there's a lot of variability, and you super smart people out there that have on the ground knowledge might think that you're better or worse than these averages. So feel free to change them as you wish for any individual lake or all lakes in your scenario. Now, I mentioned too that we talked with a bunch of counties and asked them about the average cost for each one of these interventions. And these, again, are estimates, and we know that they vary between lakes and busy days and slow days. So please go in here and change to your heart's content what the cost is for any one of these interventions. And it adds it up for you based on the number of boats that you're going to come in contact with, what that estimated cost is for your scenario over a 1 year period, over like a field season. So you can see in this case, if we do some fairly intensive watercraft inspections and decon at 5 lakes in Crow Wing County, and we do education everywhere, it costs about $183,000 to make that happen. Now, this is interesting information about just sort of what things look like, but you can submit this. This is the last step of the tool. You submit it to run the models. Like this is where the power of the algorithms comes into play. You just push submit, and all this information you've entered into the pipeline will run behind the scenes. And because these are so huge and computationally complex, unlike the other aspects of the AIS Explorer, this one takes a little bit more time to run. So you will put in your email address, and it will email you a report in an HTML format that you can browse through. It shows you some interactive features about how well this scenario performed compared to the status quo. So just as an example, and this is a different scenario than the one I just showed, but as an example of what it might look like for you as you go through it, these are the main sort of outputs, and there's a lot of other information in there, but this will show you the lake level change in introduction risk across the state or your county of interest, and the number of infestations averted based on that management strategy. So in this hypothetical scenario, the number of new infestations that we would predict to happen, assuming no intervention, is something like 18 new lakes will become infested in 5 years. If you do these types of interventions and it costs this amount of money, you could expect, based on these models, to have about 3 or so lakes become infested. So for the first time, this allows you to look at that cost and the benefit of your management strategies for any number of scenarios that you want to come up with. You have one great idea, put it in there and see what happens. If you've got 50 great ideas, put them all in there and see what happens. And that maybe for the first time will help you have some numbers to those plans. Maybe a good communication tool, maybe justifies additional investment. You can play around with this all you want. And my final slide here, as Amy mentioned, there's a lot going on. This is by no means, these are not static tools. We are continuing to invest in the development of them. We are writing grants constantly to add complexity or realism. We were talking to many of you to sort of get feedback and how you use them and how they could be changed to be more helpful. So stay tuned as more comes out. And there's my email address. If you've got questions, happy to chat more at any time. Always excited to get the feedback. And I will just mention quickly, I'll drop into the chat here a recording of a presentation that walked through how we quantified the effectiveness of those different preventions and the cost. I suspect there'll be some questions on that. I don't have the time to go through it all, but I'll drop it in the chat and we can follow up later if you have got more.
Yeah, thank you. Thank you, Nick. I know these are kind of like teaser presentations so that people can go to the websites and learn more, play around with the Explorer tool. So thank you for that. Do we have one quick question for Nick before we move on to our next speaker? Mike, go ahead.
Yeah, I was just wondering if you found any interventions that are like the highest cost effectiveness, in some of this work.
Yeah, Mike, that's a great question. And the answer is it really probably depends on the dynamics of the network and the system you want to work with. So in areas where you have a lot of infestations, you have to spread out resources, and it's probably a cheaper option like education where you can cover more ground. If you have few infestations, I'd suspect, and we can test these in the models, more targeted, like more intensive intervention, like inspection effort or decon might be more appropriate. Sort of isolate those infested lakes. So it depends on your strategy, your values, motivations, what you want to get out of it. It's probably going to change the answer to that question. But you can play around with the tool and see for yourself for your area.
Thanks, Nick. John, do you have one more quick question for Nick? We're working to unmute, I see. And I can, no, I can't unmute you. All right, maybe we'll come back if we have time after Alex's presentation. We do have about 50, go ahead, John.
Yeah, I'm sorry. I finally figured out the controls on this thing. Anyway, excellent presentation. I put in a little question in the chat. So it's a fascinating tool. I played around with it for our little lake, Spider Lake in Nevis, and if we did 100% of boats, inspected 100% of everything, it would be like $1.5 million a year. So it's like amazing. But then the opportunity cost is that that's still about 1% of the cost of trying to treat the lake. So the other thing I would love to see is that there's a window of opportunity here. Prevention is cheaper. It's still expensive, but it's much cheaper than the future. So playing that in, it may be really very important right now. There's a surplus, a bigger surplus in the legislature. Getting more prevention done now is going to be much more cost beneficial than waiting to try to treat the lakes, which isn't as successful. Thanks.
Thanks, John. All right, I'm gonna kick it on over to Alex for our last AIS Research Center presentation here.
Yeah. So I'm not as familiar with I shared the wrong window here. See, that's what I mean. I'm not as familiar with Teams, so give me 2 seconds here to
That's all right.
figure out which.
We've got a good 15 minutes for you still. So
Okay, good. I
yeah.
just have 2 windows with the exact same name open, so I couldn't figure out which one I was supposed to be clicking. But you all can hear me and you can see my screen now.
Yep.
Okay, perfect. So thank you everybody for having me here. I am Alex Badges. I'm the quantitative ecologist on staff for MACERC, so think data science. And I'm going to be talking to you guys today about another one of our apps, a different app with a different purpose, but similar theme. I will say that my job here at MACERC is to sort of facilitate all things that our center does with respect to data. And this includes advising on specific projects, but it also involves sort of overarching initiatives that enhance the work of MACERC overall, as well as that of our partners, to sort of bolster our collective understanding of our state's waters. And that's why in late 2022, MACERC tasked me with creating PI Charter, an online database and dashboard to collect and host PI or point intercept aquatic plant survey data, following on work that was conducted for many years by Dr. Mike Verhoeven, who had been collecting and aggregating these PI data from various sources around the state. This doesn't really do it justice, but I'll say that PI Charter sort of exists to catalog everything we know from PI surveys, at least, about the aquatic plant communities that inhabit our inland water bodies. This is useful for asking AIS questions, especially when you get it all in one place. And that's why MACERC is interested in taking on this task. But we also see it as an all-around reference tool for understanding our state's ecology. I mean, aquatic plants are understudied, and so having this as a giant resource to understand them better, we think is really important. And I'm here talking to you guys about this today. I actually have talked about PI Charter to this crowd before. I'm here today specifically, though, because I just spent 2 months overhauling PI Charter to what I'm calling version 2.0. So I'm here today to show off that new version and also to seek feedback on it. There are 4 tabs on PI Charter, but I'm really only going to be focusing on the overview and records tabs here because I think these will interest present company the most, and because MACERC, and in particular me, want feedback on these tabs from you all in particular. Are they doing the right things? Are they doing them well? What more could they do? I'll be frank, I'm the quant guy behind this app. I programmed it and I designed it. But while I do hold a PhD in plant ecology, that doesn't extend to aquatic plants much at all. So I'm really counting on the input of experts like you all to make sure that this app is everything that it should be. So yeah, I'm not going to be using slides today. I'm actually going to just be live in PI Charter, showing off some of the features, pointing out places that we'd like some constructive feedback. I hope it won't make a fool of me by doing this, but that's always the risk of trying to do stuff live. But you can actually follow along with me live if you would like to. You can go to PI Charter on the web using that link that I just dropped into the chat. So if you want to follow along, you're welcome to. I suppose we could crash the app, but I would consider that to be a win if we did. So feel free to hop on it if you want. I'm going to be starting, I lied a little bit. I am actually going to start on one of these other tabs. It's the leaderboard tab. This is where we acknowledge the hard work of the surveyors and data submitters who built our database. Without them, none of this would exist. So you can consider this to be my acknowledgement slide, if you will, for this talk. And you will see that the most common surveyor is an MNDNR surveyor, and that the four MNDNR programs that do PI surveys are amongst our most common submitters, and that's not sort of an accident by any means. I have heard from the MNDNR that they want all of their surveyors anonymized, which is why you're not seeing any names here. I'm trying to honor that request as well as I can here. I'm still not actually sure if I've done it the right way though. So if anyone has any opinions on this, or if you think this is sufficient, please let me know. I'd also be happy to build in some kind of opt-in out, opt-out mechanism here if that makes sense. But I wanted to acknowledge the important role of a bunch of people, including many at the DNR, in building this tool. With that addressed, I'll sort of bounce back over to the overview tab, which is where you land once you open the app. What this tab shows is at a glance all the lakes that we have records for, each marked with a dot on the map. If you hover over a dot like this, you will figure out which lake it is. So this is Norquist Lake up in Lake of the Woods County. And then if you click on that marker, it'll turn purple and you'll get access to even more data still, because a summary of the records for that lake will appear over here in this table on the right, where they can then be searched and filtered and sorted and all that good stuff. You'll see how many surveys we have, when they were done, how many taxa were found, and which taxa specifically those were. Now, this year, something new, you'll notice that some of the markers are in green, and if you highlight over, you'll see why they're in green. Even when the data weren't gathered by the tribes themselves, if the lake is on or adjacent to tribal lands, we are voluntarily now redacting those data from display to respect tribal data sovereignty. And there's more about this new policy down here in the disclaimers section on this tab, which is also new. Speaking of which, there's also a disclaimer up here about anonymization of MNDNR staff when it comes to surveys. So you can read more about that there and let me know if I've honored those requests adequately. And then you'll also find a disclaimer about protected species. We've used a list of species provided to me by the MNDNR to anonymize our data for specific protected species. For example, you'll notice at Norquist Lake, there's a protected species 6 that has been noticed there before. I think the way that I'm handling these protected species is meeting all the relevant statutes, both federal and state, but I'm no expert on this either. So if anyone has feedback on how I'm handling this, I would be welcome to have that feedback. To quickly highlight some other features of this tab, you can filter this map in several ways. So let me just clear everything for a second. If you open up this filters and toggles menu, you can filter this map by a specific lake if you would want, or a specific county. These are also search boxes, so you can back out things and type in here if you would like. There we go. So I can select a county or a lake that way. And what will happen is all the lakes will come up that are just within that county, for example. You can then select all the markers, except for the tribal ones, all at once that are shown on the map using this button. You can then click these two buttons over here to get a list of all the lakes that are currently being presented in the table, or all the taxa that are currently being represented in the table. These are in copy pastable form. If you want a copy of the table here, you can download it as a CSV file, which you can open in Excel. And then you can clear any of the selections that you've made up to that point by hitting this button here. Two particularly interesting features, though, that I want to highlight. This button, which I've added this year by popular demand, will toggle all the names that are currently scientific names on the app to instead common names. So if I click that button, you will notice that if I go to this drop-down menu now, all the species names or taxonomic names are now in common name form, So if I hit the button again, they return to their scientific names. So if scientific names aren't your thing, this might make the app more consumable for you. The other thing you can do is use this to search for specific taxa. So if I pick some taxa like this one here, what will happen is the map will actually filter to only the lakes where that specific taxon has ever been observed before. So you can see that this particular taxon is relatively rare. So I think this is a cool way of sort of exploring the geographic distribution of different taxa as well as their rarity. Just keep in mind that less than 10% of all Minnesota lakes have been surveyed based on our records, and most of those only once. So many taxa are probably more dispersed than we know. But this is still a cool way of exploring those data. The last thing I want to show in this tab is this feature. It's the Start Here menu. If you're new to PI Charter, this is a great place to start because there's a bunch of useful info in here. There are tutorial videos in here. There's history about the app, what a PI survey is if you're not familiar, why we think they're valuable and we're collecting them, and so on. Maybe most newsworthy, though, is this last option down here. In this section, you'll find template contract language that we're hoping organizations will use whenever they contract for a PI survey, such that the submission of those data will be included as part of the contract. So we want feedback on this language from you all. It's our first attempt at trying to do this. We've had it requested of us in the past. And we also would love your help in amplifying or using this language if that's relevant in your context. And you can also find this language on our MACERC website as well. So that's the overview tab. Our hope is that it will be a useful way to quickly see statewide diversity data on aquatic plant communities and assess where our understanding of them is maybe incomplete or misaligned. I'll now just briefly mention the submissions tab, which I completely overhauled from scratch in this version. The goal of PI Charter is not just to house and display PI survey data, but to gather them too. While the MNDNR performs more than half of all the PI surveys in the state, many other organizations perform the rest. We're talking park districts, consultancies, lake associations, watershed districts, counties, and more. And it's our goal to bring all of those data together into a single database. And that requires having a submission system that can catch those data, their metadata, clean them up, and combine them into a unified form. And the submissions tab does all that using just a max of 19 very brief and easy to answer questions. It's kind of like a Google form, if you will. I'm very proud of it. You can play around with it if you would like to. I'd welcome feedback on it, but it's too complicated. So I have to skip over it right now. And instead, I want to end on the records tab because this tab is both new and still under construction. So it's a place I really would like some feedback. The idea of this tab is for it to be sort of the anti-submissions tab in the sense that this is where you're retrieving the point level data instead of sending them to us. You can do this by first selecting a lake down here. So I'll select Susan Lake because I know we have a bunch of data from Susan Lake. So this is a good place to start. Let's see here. Okay, it's lagging on me. So instead of doing that, I'll just pick any old lake here. And then what you'll get once you pick a lake is a list of all the different surveys we have from that lake, sorted by date. If you then click these, and if there were multiples, you could click and highlight multiples. What the app will do is download those particular records and then display them over here in a table on the right-hand side, where you can then also download those records if you would like to. You can, so what this tab I hope demonstrates is that we're not looking to take the data and hoard them, but make them available to everybody who wants them in as consistent a form as possible. A couple of caveats here again: protected species won't be displayed. Nothing on tribal lands will be displayed. If the survey was conducted by a DNR program, the surveyor's names will be redacted here. The submitter's email address will be redacted. The other thing I want to point out, and this is another disclaimer down here, is we do perform some QA/QC on submitted and aggregated data to ensure that their logic's internally consistent and that we're not introducing any data entry errors or there aren't any obvious data entry errors. But we're not warranting these data. They are as is. We're trusting that the surveyors knew what they were doing when they conducted the survey, and we're just trusting that these data are good from the outset, basically. And there's a policy that talks about this down here. So we can talk more about that in the Q&A if you want. The last thing I will mention is that this tab should do much more than it currently does now. I want it to be displaying over a series of subtabs here, a bunch of useful visualizations and summarizations. For example, right now it's just a test case. The app already calculates percent cover, both by survey and by taxon. So there's only one survey listed here, but you can see all the different taxa are listed here as percent cover. I want there to be maps and graphs and tables and all sorts of useful things here, but I'm not a subject matter expert. I simply don't know what all this tab could or should be doing, but I'm really open to ideas. If we can make this tab a powerful exploratory and fact-finding tool for you, or even make it something that saves you time, we want to do that. So I'll go ahead and stop sharing here and wrap up. And then if there are questions, I'm happy to address them.
Thank you, Alex. Yeah, we have a few questions if you have, or a few minutes for questions if you have any for Alex. John, go ahead.
Okay, and I figured out how to work the microphone. Yeah, do you have a print function on your app? I can't find it.
Print. You mean like you would want to print the screen?
Right. So I found the species, the taxonomy, you know, they got the taxa there, but I don't know how to download it, or I don't know how to print it out. Is there a way to do that?
Yeah, on a number of the tabs here, let me share. I think I can answer your question here if I share my screen again. Oops, I shared the wrong one. I should just close that one so that I can't grab it. Like I said, I kind of wish they didn't have the exact same name. Okay, so on this tab, for example, you can download the data that are in this table by hitting this button. On the overview tab, meanwhile, in the show hide filters, if I zoomed out enough to actually end up with any taxa on here, I'll just select the whole table. You can download everything that it's in the current table by hitting this button. I'm thinking you're probably talking about like this list of taxa. This there isn't necessarily a way to print, though. If you do right click, you should get a print option. What my envisioning was here is that you could copy and paste this into something. So if I copied and pasted all this into, say, Word, it would be easy to print it there. So I made sure that these modals, which is what they're called, these pop-ups, were copy and pastable so that you could easily grab the information that was on them. Does that answer your question, John?
He's nodding yes.
Okay.
All right. Any other? Yeah, Alicia.
Thanks. This has been really interesting. Somewhat of an idea for your the last screen that you shared, the records page, was like an idea of species richness and like what the value of each species was. You've potentially thought about that one, but is that a plan for the future to show how, you know, valuable a species is compared to its abundance in the lake and that sort of thing?
Value strikes me as something that would not be objective to measure, but certainly species richness and evenness and diversity, you know, it's not difficult to calculate, say, Shannon Weiner's index or something like that. So any of those measures, if they would be interesting or intuitive, those are really easy to get on here, and I just need to know what all they are. And so I'm definitely searching for ideas, and maybe highlighting species that are or taxa that are relatively rare might also be good, like starring any species or calling attention to any species whose percent cover is, say, less than 10% or less than 5% or something like that, or only appeared at one location, might be a way of sort of calling attention to, hey, here are these somewhat unusual or rare taxa that are appearing in this survey in this lake.
Yeah, I think there's another index that would take into account, like, curly-leaf pondweed and milfoil are assigned low values because they're invasive, versus a rare species is assigned a high value. And I know there's accepted indexes with it. I'm not the person to tell you what they are, but it would certainly be interesting. And there's multiple different ways to do it.
Yeah, sounds exciting. I'd love to do it if someone is willing to help sort of guide me, shepherd me. I can definitely set up the code to calculate it, but I don't know that I'd be the right decision maker to decide exactly how to do that particular calculation, but it sounds really cool and useful.
Thanks. Any other burning questions you have for our morning presenters, Amy, Nick, or Alex?
Just one other question. Alex, do you have a link showing just pictures of the taxa? Most of us aren't really familiar with the, the actual visual presentation. Thanks.
Yeah, I hadn't actually thought about that. So I will say it was a huge undertaking, probably at least a week's worth of full-time coding work just to get the common name scientific name swap worked out. Because I had kept getting that feedback and I really wanted to deliver on that. But I had been thinking about how wouldn't it be great if a common name or a scientific name was actually a link and it just threw you over to Google. Because if you could easily click on something and say, oh, and it takes you over to Google Images. The only problem is obviously we have no control over what shows up on Google. So it could show you the wrong taxa. I suppose if I went in and created a database of here's a really good link for every taxon, it could be that link instead. That thought had never crossed my mind until right this minute, but I'm intrigued by it. I'll have to find the time to do it. But especially like Minnesota Flora is a really good resource that might have good, reliable pictures. Yeah, Minnesota wildflowers would be a good choice. That doesn't feel like it would be too onerous. So if maybe I could even make it be in a pop-up actually, and what came up with just a picture of that taxon. I'd have to think about where to put it on the app, but it's a fantastic idea and I appreciate that feedback.
Thanks, Alex. All right, it's about time for our break. So we are going to take 5 minutes and then get back to our topics at 10:40 with Mike Belinsky talking about our permanent decontamination station. So everyone take a 5-minute stretch break, get up, move away from your screen, come back at 10:40. Thanks, everyone, and thank you to our morning speakers. All right. People getting back into their seats here. And I'll just introduce our final topics of this afternoon. We got 4 more speakers for the next hour and 20 minutes or so. And then, yeah, again, we'll have time for presentations and try to leave a little bit of time for questions and answers after your presentations as well. So we'll start off with Mike. I see you there. Hi, Mike. If you want to start sharing your screen and kick off your presentation, let's keep on trucking.
Sure thing. Is my screen up there? Can everybody see it?
Yep.
All right, excellent. Well, hi everybody. My name is Mike Belinsky. I'm the Watercraft Inspection Supervisor in Region 1. So I'm based out of Fergus Falls, but I cover all the Northwest region. So I run from Fergus Falls all the way up to Lake of the Woods and Red Lake area. So I'm here to talk about the permanent decon station that we installed at Red Lake last spring and into the summer. So before we get too far, so upper and lower Red Lake, they're located kind of way up there in the northwest corner of the state. It's about a 4-hour drive from Fergus Falls for me to get up there. I have staff that work out of Bemidji, and we've been transporting our decon unit and stuff up to there from Bemidji. So now that we have a permanent station up there, they don't have to drive a big trailered unit up there quite as often. So just some interesting facts about Lower and Upper Red Lake. Lower Red Lake is the 2nd largest lake in Minnesota. Upper Red Lake is the 4th largest lake. It's actually one big lake connected together, but Upper Red Lake is kind of sectioned off for non-tribal use. The rest of the lake is all for tribal use only. And it's the 16th largest lake in the United States. So it's a pretty big draw for us in Minnesota. It's a terrific walleye fishery. The crappies are doing quite well up there. It gets tons of traffic. If you go up there on fishing opener and try to find a parking spot, you should bring a walking stick and some extra water, because you're probably going to walk 2 miles to get back to the access after you park your boat there. So it gets a ton of traffic. So with that traffic, perhaps it was inevitable, but we certainly did our best to inspect up there. But there's a lot of, there's only 2 public access, and there's also several accesses on accesses at resorts. And in some of the harbors of the resorts there in 2016, we found starry stonewort. We've done some treatments up there to kind of knock it back, but it's still persistent up there. And then in 2019, through water sampling, some zebra mussel veligers were found. To my knowledge, I don't believe we've found any adults yet, but the veligers are. I don't know if we found veligers recently, but I know we did find several at one point in time. And then not too far away, up on Lake of the Woods, there's also spiny water flea that is a threat to the lake as well. So with that, in 2021, late summer into fall, the legislature appropriated $300,000 for use to prevent or to address aquatic invasive species in the upper and lower Red Lake area. This became it was obviously a priority of the legislature since they got it passed. So that became a DNR commissioner priority, and it was also a very high priority for Red Lake Nation as well. So the breakdown of that money was roughly $110,000 to Red Lake Nation, and then $190,000 to the Minnesota DNR. The one caveat being that the funding had to be used by June 30th, 2023. Now it seems like a long time, but if you remember way back then, the whole world was just coming out of a big shutdown, and it was very difficult to buy a lot of things in any timely manner or find people to work on them in any timely manner. So we worked with Red Lake Nation to see what some of their interests were and what kind of things they wanted to do, and decontamination was a very high priority for them. We worked with Beltrami County as well a bit, and then through this project inside the DNR. So the watercraft inspection program is in the Ecological and Water Resources Division, but we also worked heavily with Parks and Trails for development of the of all the engineering and the site coordination and stuff like that, management resources to get info on the stuff we want to build, electricity, that sort of stuff. We worked with community communications and outreach to get the word out there that we were building this. And then we also had the commissioner's office for help getting us all this stuff pushed through in time for the deadline. So with decon being the kind of the high priority, Red Lake Nation decided to install 2 CD3 machines within their tribal area. We also drafted a bunch of AIS messaging inside the fishing regulations handout. We got some Swedish dishcloths made, and we did a lot of outreach at Big Bog Days, where there's over 700 area students that come and check out the park and learn about all things natural resources, AIS being included in that. And then some Facebook posts that got close to 200,000 impressions, kind of detailing some of the work that we've done up there. And then, of course, the main topic here was the permanent decontamination station that we got built. So decontamination stations, so there on the left, you can see the typical one we've had in Minnesota. You probably recognize, our state truck and our yellow trailered units. We have a couple that are not yellow, but generally the trailered pressure washer units that can deliver high heat in a big mat. You can contrast that to, something like what Utah has over there. It's got the big trailer and then the big green pads where you can drive up. I actually used to work for Utah, and I had one of these, and while they look nice, they also had many problems as well. I would say that our trailer units are every bit as good as those. I did like the permanent mats that they had. Those are very nice, but I think our trailered units work just as well, if not better than those in Utah. And then Colorado, this is their kind of version on the right there of the permanent decontamination station. It's not a glorious looking thing like Utah's got. It's just a little shed, but it packs a punch when we want to kill some zebra mussels or stuff off of boats. So we decided to kind of follow that Colorado model of what they built. And so first came site selection. That sounds easy, but this was probably a 3 to 4 month process trying to get that figured out. We kind of knew that we wanted to put it at the Homestead access over there in Wasatch. But to do that, first we had to figure out who owned the site, which you'd think would be easy, but it was not as easy as it seemed. A lot of stuff happened on paperwork before computers, and nobody really knew where that paperwork was. So it took a little bit of time to find it. Once we found it, then we had to go through and see if there was any archaeological stuff that was needed. Fortunately, we avoided most of that. That can be a very time-consuming process. But since the access was built there, a lot of that archaeological stuff was done already, so we knew what was in the area, and we could kind of speed that process along. Next came the design phase. Everybody kind of had their own ideas, so it was kind of getting everybody together to settle on some ideas that would work for everything that everybody wanted to happen up there. And then construction, we were going into winter, and the winter was not as friendly as this winter. So actually we were concerned we weren't going to be able to get in there and pour concrete as very early in the spring to hopefully have it up and ready for fishing opener. So we laid a bunch of straw down. Some of our Parks and Trails partners, they took a part of a day and brought in a whole bunch of straw bales to kind of insulate the ground, keep it warm, so it would thaw quicker and we could get that concrete poured right away. And thankfully that did work. It probably wouldn't have been an issue this year. You could probably pour concrete right now if you wanted to. But we battled through that and we got that installed. But each step along the way, all had its significant challenges to overcome, and that takes time. And if you remember, we were up against the June 30th deadline, and we certainly cut it very close, but we were able to get it done. So here's an overhead view of the homestead access on upper red there. So that gravel area, it does get some parking in it. So we kind of took over some of the parking, but we didn't want to have to mess with any of the parking spaces in the paved area there. That's one of the regulations that you kind of have to go have a hurdle through when you're building stuff on public water accesses. You can't impact the parking that's already there based on the fact that there's probably some federal dollars involved in constructing the access originally. So part of the cost was kind of redesigning this access so we didn't impact parking at all. And you can kind of see a little final view of the engineering plans there. So there you see our rendition of what Colorado has did. It looks pretty basic, but and it is. It's an 8x10 shed. We've got some well water that was run. We had to drill a well. The old well that was there was not functioning very well. It didn't have enough water output. We had to run electricity there. We had to get propane installed. And then inside there's 2 on-demand water heaters. We have the concrete pads that we poured. There was also some site grading, curb and gutter that needed to be put in, some seating, stuff like that. And we still haven't quite finished off some of the traffic signs to kind of show traffic flow. We didn't get to that year, but that's up next for this year. Get some stuff put on the pavement. So here's the inside. This is kind of what everybody wants to see. So our trailer units are fairly detailed, hard to work on, and then you see this thing, and it's actually pretty simple. It's 2 on-demand water heaters. They're tied together in tandem, so they heat the water up to 140 degrees, and the water will stay at 140 degrees the entire time that you're using it. It doesn't dip in temperature like the trailer units do. So it's really a pretty simple setup. The only thing that's missing here is a pressure washer. We're adding that this year. The pressure washer really is only needed to remove stuff from boats. It's not used to kill anything. You don't kill zebra mussels with it. So we're just adding a simple, little rollaway one that you can get at Home Depot for, 150, $200. Just something cheap and easy to use. You can tie that into there's a hose bib on the outside we can tie it into and use it right there very easily. That was a cost savings over installing a permanent pressure washer in there. That was a couple more grand on top of it. So we decided to go this route since we don't need the hot water to go through it. Another question we hear a lot about is the cost. Anytime the state builds something new, it's going to cost a lot of money. We've never done it before. It takes a lot of designing and a lot of engineering and lots of hands involved. So the cost of this unit was roughly around $209,000, which is a considerable amount. But we had to drill a well, we had to pour a lot of concrete, and as everybody may remember, coming out of COVID, everything cost more. So there was nothing cheap about it. But so like I said, we're going to work on signages. We're actually going to try and partner with Beltrami County. Their AIS guy is retired this last year. Used to be a former coworker of mine, but when they replace him, we're going to have a meeting and work on setting up an MOU so Beltrami County staff can also use this, so we can kind of maximize the amount of time this thing is up and running. We're going to continue our education and outreach efforts and continue monitoring for AIS up there. The big question is how can we use this design for other sites in Minnesota? And now that we have this design, how much cheaper can we make it? Because obviously, we can't go around installing $209,000 units everywhere. So the cheaper we can get them down, the better. We're pretty convinced we can get this down a considerable amount, but we'll have to wait and see. I think we're working on building some other ones here that are at least in the works. I haven't heard a lot of details on them yet, but hopefully we'll be adding a few more in the state in the near future. Any other, I guess we got some time for questions here.
Yeah, we
Anybody
do.
has any?
Thank you, Mike.
You bet.
Any questions for Mike?
I see there's a question on building offsite locations. I don't see why not. Obviously, it's always nice to have it right on the axis where the boaters are. There's always the challenge of getting boaters to drive to an offsite location. That's not to say that boaters won't do it. We know that they do. They are willing to do it. So, I would say there's certainly always room for more locations on or offsite to anywhere that's easy to get to from some high traffic lakes.
All right. Well, thank you, Mike.
You betcha.
So next we have Courtney with the EPA out of Duluth. Courtney, are you there?
Yes, let me share my screen.
Wonderful. All right, looking good.
Perfect. All right, well, thank you so much for having me. I know we're talking about new approaches, new technologies today, and eDNA is definitely one on the top of all of our minds with invasive species. So I'm going to give a bit of an overview of what this means for invasive species management and detection, and then dive deeper into one case study of how we've used it in order to get you thinking, hopefully have a bunch of time for questions. But especially, this has been an ongoing project with lots of people at the EPA that I definitely want to acknowledge the whole team that's been a part of this, even before I started working at the EPA. So when we think about an invasive species introduction and spread, this is the model that we use for whatever ecosystem or whatever invasive species, that you have this new species, gets to a new place, it becomes established, population grows, and it then can spread to new ranges. This is what you've seen a million times in your own systems, your own lakes, and your own species that you're concerned about. But of course, most of the times that doesn't happen. They're not introduced, they're not established, they don't spread. And those cases are also really important because it helps us with our management and our monitoring strategies. And so when we're monitoring, it's, as you know, important to intercept at the early stages when they're easier to eradicate and before there's ecosystem damage. But this is really difficult with the conventional surveying methods. It's really, as you know, time consuming, lots of samples, and the taxonomic expertise can be really, really advanced. These are needle in a haystack situations. And this is where eDNA is a new method that could come in to help make this process more efficient. So when we say we're using eDNA for invasive species detection, eDNA is the DNA that's in the environment that's shed by all sorts of different species, including invasive species. So just by being in the environment, living, eating, pooping, reproducing, all of those cells with DNA inside of it get shed into the environment. And then you can take a water sample in order to collect that DNA, and that DNA collection can then be isolated through filtering. We actually do this a lot of times just at boat launches. So we'll take and we'll collect the water out in a lake, bring it to a boat launch, and have this like mini, almost forensic lab where we filter out the water for the eDNA. Then we take those filters, we bring them to the lab, and we do qPCR. And that's this analytical technique that allows us to see whether that DNA target is there or not, and in what relative amount, and kind of see whether there's the invasive species in that site. And if this all seems really familiar, that's because this is what they did for COVID detection in wastewater monitoring. So, you know, you would hear about how we've got high levels of COVID in our wastewater, so there must be a lot of transmission in this city. It's the same exact thing that we're doing for invasive species. So it's like saying there's a lot of invasive species in this lake, just like they did for COVID. And so I don't want to get into the weeds about all of the technical details that are involved in this, because I think this is where you get really good partners to come in and collaborate on all of those details, because a lot of times they differ from project to project and from system to system. But this is the general workflow that we do when we do eDNA, implementing eDNA into our invasive species projects. And so I'm going to do a case study, and the take-home messages I want you to think about during this case study is that eDNA surveys provide complementary information to the conventional methods in the early detection, monitoring of invasive species. So this is a complementary approach. And that methods can iteratively build on each other over multiple surveys as technology develops, as management concerns change, and as research questions arise. And so when we're building these new approaches, this is an iterative process. It's not a magic on switch. It happens over time, over collaborations, and as the systems and the technology changes. So I'm just going to dive deeper into one case that we've used in zebra and quagga mussels, or Dreissena as they're lumped together. You're already familiar with these. They're invasive mussels. They're a huge problem in Minnesota. And what's important to know when we think about eDNA with these is that there are veligers that disperse into new habitats in the pelagic zone, and that there are adults that settle onto surfaces, and eDNA is contained in both of those life stages that can be collected. And that there are two species that are in this, the zebra and the quagga, but sometimes they can even be a little bit hard to identify. The veligers actually can't be identified to species. You can just know that it's a Dreissena veliger, where the adults can be identified to species. And they occupy a little bit different habitats. So their population dynamics can be slightly different, even though we lump them together. Okay, so the system we're working in for this case study is actually Lake Superior, which I know is pretty different than a lot of the lakes and rivers you work in, but it provides a really good system for us to study this invasion front. So we generally think it's inhospitable to zebra and quagga mussels. It's too cold, not enough calcium. But the St. Louis River estuary was invaded in 1989. We do have a large population there. And there's been scattered individuals collected throughout Lake Superior over the years, but particularly in 2011, there were a few individuals found in the Apostle Islands. This is a National Park Service site, so it was of conservation concern. It raised some alarm bells. And so the first management question that we had, that we started to think about eDNA use, was what is the distribution and prevalence of the zebra and quagga mussels in the Apostle Islands? For more information on the methods and the study, it was published in the Journal of Great Lakes Research for additional details. So in 2017, where we stood on methods to do a survey of Dreissena were to look at adults on the bottom, to look at veligers for zooplankton toes, but we added eDNA. But at this time, where the technology sat was we could only do a genus-specific qPCR marker. So it was just telling us zebra or quagga are there. And actually, it didn't work. We didn't get any positive hits for Dreissena on the Apostle Islands. The only thing we got was veligers had some zooplankton toes. We did find veligers. And this map shows the veliger distribution in the Apostle Islands. And it shows this interesting pattern that from a management perspective, it was really interesting, because it looks like the majority of the detections are on the western side, which is closest to the St. Louis River estuary and perhaps where currents may be flowing. So we started to think, where where are these coming from? This isn't associated necessarily with boat launches, but actually with being closest to the St. Louis River estuary. So is this perhaps a current transport to get the mussel veligers into the Apostle Islands? So this was kind of our second phase management question is, do we need to be worried about currents in Lake Superior transporting these? And for more methods and results, and dig deep into this, again, was published in this paper for more details. So now, 2 years later, 2019, we actually have a better eDNA marker now. It is species specific. It can tell us whether there are zebra mussels or quagga mussels there. So we have a better DNA tool. And we also, because we were looking at current transport, we didn't look at adults, but we were doing veliger tows, as well as looking at abiotic factors that could affect the survival of these veligers. And these map on the left shows the points we sampled. And so this is the only graph I'm going to show, but I think it really demonstrates how just a few years as the technology develops, how you can improve and get better results and more interesting results. So on these graphs, the two panels represent the traditional kind of conventional sampling of veliger tows, and then the bottom panel are the eDNA results. So we did find from the spatial gradient on the x-axis from the estuary at the farthest left point to the Apostle Islands on the farthest right point, a decrease in veligers. And so this is really good evidence that there is currents that are transporting these mussels to the Apostle Islands. But look at these eDNA results. This shows a very similar pattern, but it tells us a lot more information because now we know that there's both zebra and quagga mussels that are along this pathway. That's really interesting. We would have thought it might be all zebra because that's normally what we associate with the estuary, but no, we do have both zebra and quagga. This is really interesting. But also it's more sensitive than the conventional methods. There were 3 samples that did not turn up any veligers in our first look at the sample. They got a positive eDNA hit, and so we did a research. And when we searched those samples a second time, we found as low as 1 veliger in that entire zooplankton tow. And so the eDNA was actually doing a better job of detecting these than the conventional methods, which is really shows that it's a good tool. So because now we know that the currents can transport Dreissena, what about all of Lake Superior? Maybe this isn't just the South Shore that's at risk. The eDNA allowed us to say, maybe we need to further monitor all throughout Lake Superior, which is at this latest phase. We did undertake a full survey of Lake Superior using the species-specific qPCR, as well as adult and veliger surveys. And this time, we actually did find evidence of zebra and quagga mussels in all of our sample types. And that tells us that as we're iterating back and forth in these surveys, our tools get better. And perhaps, yes, there is probably more, there's probably spread of Dreissena that's happening. There probably is more of the zebra and quagga mussels in Lake Superior. But also, I think we're just getting better at our tools as we try these new methods. And so what we found just briefly in Lake Superior was that the conventional veliger tows, we only found them in 3 samples throughout Lake Superior, as shown here in red. All of these white X's are no detections using our traditional Great Lakes National Program Office method. But here's our eDNA results. There's a lot less X's of no detections. We're finding a widespread eDNA signal of both zebra and quagga throughout the entire lake. And this is pretty surprising. This is the first time there's been a signal of zebra or quagga in the middle of Lake Superior. And so this tool is providing new insight in how we manage for zebra and quagga mussels in Lake Superior. So we can conclude that the eDNA is more sensitive than the conventional methods, and that there's a different pattern in zebra and quagga mussels, and so perhaps there needs to be more species-specific investigation of these 2 species now that we have the possibility of a genetic marker to differentiate the veligers. So let's go back to this graph I showed you in the beginning, and I hope that you can visualize your own system within this. But I'm going to go back to what we found out in the context of zebra and quagga mussels. So we know that there's established populations in the St. Louis River estuary, Thunder Bay, Nipigon Bay, that these are established adult populations that are reproducing. They're pretty extensive. But we also have some populations that represent spread, but are pretty well managed. They're actively managed and removed. And so some years they get more detections than others, but they're pretty small populations. They kind of represent the spread to new range. And the methods we use for these are adult surveys, looking at actual adult mussels, and that shows us where these populations are. And that type of a detection is really important in order to manage, so we don't have this cycle right here where the spread to new range becomes established populations. But then we also have these cases where we get veliger detection or eDNA detection that represent they're not spreading. So I don't think there are any adults that are in the middle of Lake Superior. These don't represent populations that are reproducing. This actually represents a risk pathway of where we know that there's opportunities there, where if there is environmental change, perhaps, or if it's the right time, if there could be populations in the future. And so this is really important for risk assessment. And so the way we're thinking about these eDNA positive hits are actually that the eDNA is illuminating a risky pathway where further adult monitoring is needed. Because now we know that there's this place where they're spreading, there's evidence of spread. If there's correct habitat there, that's where there needs to be really active monitoring for these adults, so it doesn't become one of these kind of spread to new range type of populations. So perhaps within this framework, you can think about how in your system and your species, this eDNA could fit in. So again, take-home messages. eDNA surveys provide complementary information to the conventional methods in early detection of invasive species. This was the case of the veliger and the eDNA toes for Dreissena, especially like in the middle of Lake Superior. This is complementary to those adult surveys. And then second, that methods can iteratively build on each other over multiple surveys as the technology develops and the questions change. We did that from kind of a crappy marker the first time we went at it that didn't detect much and didn't really add any information to one where we're actually getting a lot more information from it about species-specific patterns. And so here are more details where there's additional, technological information about what we did and what we found in these two citations. And here's my email. I'm really interested to discuss more, happy to partner, and I'm interested to answer whatever questions you have. So thanks for letting me talk.
Thank you, Courtney. There are a few questions in the chat, so I'll read a couple of them off here. And then Doug is also responding to some of those questions as well. So a general question, how long will eDNA remain in the water?
So it really depends on the environmental conditions. It depends on the temperature. It depends on the chemistry, the amount of light, the time of year, but it can be a matter of days to weeks. But it really varies, and that's something to look into the literature of what's been found in whatever species you're interested in, and kind of pilot.
Courtney, this is Doug.
Yeah.
So some of the literature shows that the invasive carp in the Chicago River, that the eDNA can survive up to about 7 days. I don't know if that equates to Dreissena DNA, but that's some of the information I'm aware of.
Thanks for that addition, Doug.
Yep.
Another question, and you might not know, but other people on this call might. What is the cost for eDNA testing? Is it commercially available?
Yes, it is commercially available. There are labs that you can submit samples to, but the cost really depends on what species you're going for and what methods. It can really vary. Like if you have a species that's really commonly monitored for, like the carp, like some of the invasive fish, that if they already have a really reliable marker that a lab can easily implement or may even already have all the supplies for, that's going to be less expensive than some rare species that we don't even know that much about. Um, the genetics where a whole new marker needs to be developed. And so I hesitate to put a dollar amount on it. I think that's a discussion that needs to be more context specific, and getting those partnerships can really help kind of do a ballpark.
Yeah, and if anyone on the call has paid for eDNA analysis in a particular lake system, if you want to enter that information into the chat, I think people would appreciate that kind of information. Any other questions for Courtney on eDNA sampling in Lake Superior?
Courtney, this is Doug. Can you speak to the issue of detections of eDNA and the potential presence of actual invasive species?
Yeah, so I think you're referring to false positives.
Correct.
Yes. So false positives are something that they happen, but we have really robust controls in place that I think there was a reputation, years ago when this was first unveiled, that there were a lot of false positives. And since then, there's been a lot more controls that have been put in place that have really reduced the amount of false positives that there are. And so I encourage people to think think about all the controls that they can put in their study in order to be more confident that things aren't going to be false positives. But they do happen. And I think that's where having a really robust knowledge of the biology of the species that you're detecting will be important, because DNA is just a chemical. It doesn't represent the actual species. It could be a dead species. Dead things actually shed a lot of DNA, and it's not a population that represents a real population detection. And so understanding the life history of where DNA could be coming from, it could be eggs that are released or that spread somehow, but that won't survive. And that's why it's important to not take an eDNA detection as representing a living population that's reproducing and invaded a water body, but more to use it as a piece of evidence that you use in tandem with some of the life history information and conventional methods to be a detection source.
Very well put. That was very, very well put. Thank you, Courtney. One last question for you. Is eDNA only for animals? Can eDNA be done for plants or algae?
Oh, absolutely. It can be done for anything that has DNA. So just like it's done for COVID, it's viruses, it can be done for diseases. And so I know that there's been some really good eDNA assays for things like Dynamo that have been done for harmful algal blooms. And so absolutely, some of those nuisance plant and algae species have been really had a lot of success with eDNA.
Thank you, Courtney. All right, we're going to move on to our next speaker now. So Dawn, excellent. You're already on it, sharing your screen. So we're seeing the full application right now. We'll let you get to the slideshow part. All right.
Okay.
Looking great.
Looks
Go ahead,
good.
Dawn.
Great, thank you. Thanks, Tina and Doug, for the invitation to talk about this. I don't know if this is a new approach, but more of a new discovery. We may be using some new approaches, but the signal crayfish here in Minnesota, which I'm sure lots of folks have heard about. What I'll give a little background on the signal crayfish themselves. We'll talk about kind of the discovery and the situation where it was found, and then some of the response planning. what we're going to try and do about it. And whoops. Did I click on the wrong thing here? All righty. Okay. And first of all, I just want to say that besides our entire invasive species unit here at the DNR, we're working with Gretchen Hansen and Mayserk at University of Minnesota. We're working with Eric Larson at University of Illinois. Also Justin Swart, who's the Douglas County invasive species specialist. And within DNR, we're working with the fisheries folks, and I'll talk about a little more about that. And then also just to acknowledge, we've had a lot of really excellent feedback from folks like at the Michigan DNR who are dealing with red swamp crayfish and rusties. And then out west, California and in Europe, where they're currently dealing with invasive signal crayfish. And then just to give a little background here, the map on the left here shows the tan section is showing the native range of signal crayfish, which is basically the Pacific Northwest. Those kind of blue and green and yellow dots are areas where they've been introduced either intentionally or unintentionally in the Western US. And that began as early as like the 1890s. And the most recent record out west is Kodiak Island in Alaska. The map on the right shows occurrences in Europe. They were introduced back in the 1950s to try and replace a native crayfish, which had been decimated by crayfish plague. And they're now in 27 different territories. They were also introduced in Japan in the 1920s, and also intentionally. And they've caused lots of problems for one of the native crayfish there. So they're distributed in quite a few places. And then most recently, of course, as you can see, is here in Minnesota. As you can see, this occurrence record is way outside the current invasive range, so which is why it's gotten so much attention, and folks are pretty worried about it. And then just to get, so what's different about signal crayfish, say, compared to our Minnesota natives? Our Minnesota natives are in the family Camberidae, and that includes 400 plus species in North America, Central America, lots and lots of species. The signal crayfish are in the family Astacidae, which in Western North America includes at least maybe a half a dozen other subspecies and species in the same genus as the signal crayfish, Passifasticus. And some of the, so it's a whole, it evolves separately, I guess is the point here. And some of the distinguishing characteristics of signal crayfish, if you check out the photo there on the left, on the upper surfaces of their chellae or the claws, there's sort of a light bluish green, whitish patch at the claw hinge, which folks thought looked like signal flags from the railroad, and that's where they got their name. But that's a distinguished feature. Their claws and their carapace, which is the covering over the head and the midsection of the body, is quite smooth compared to a lot of our native Camberid crayfish, which are a lot lumpier and have a lot of bumps and ridges and so forth. And then when they do their defensive display, you know how crayfish, if you try to grab them, they'll flip up their claws at you. When the signal crayfish do this, as they're the older individuals, there's a really bright, distinctive color on the bottoms of the claws. So that's a good way to pick them out, let's say, if you run into them into a lake. Another thing is they're just a lot larger than our natives. Just the few which have been caught here in Minnesota, the largest one was more than 100 grams. Say a typical Minnesota native is between maybe around 20 grams. So these are big crayfish, kind of half lobster size. And then speaking with some of the folks out in California who are dealing with signal crayfish invasions, during the juvenile phase, which is between about 1 centimeter, 2 centimeters carapace length. And the carapace length is just the point of the nose, basically, to the end of the thorax. That length, when it's between about 1 and 2 centimeters, they become quite cylindrical in comparison to other crayfish species. And so they say they look kind of like walking pencils. So it's a way you can hopefully identify these things. If you're to see little ones which look very cylindrical, you might have a signal crayfish. In terms of life history and some other characteristics, the males of signal crayfish do not go through what our native crayfish do. Our natives go through what's called cyclic dimorphism, where the males will have a kind of aggressive reproductive phase, and then they'll molt and they'll go through a phase where they're not reproducing, they're less aggressive. The signal crayfish do not have those different life history phases. The other thing they don't have are what are called our hooks on the walking legs, which our crayfish use for reproduction. And then the gonopods, which are the male reproductive structures, the gonopods in the signal crayfish are basically conical and a lot simpler in comparison to some of the complex morphology that you'll see in our native Canberra crayfish. You'll see hooks and all kinds of really specific structures. The females of signal crayfish also don't have what's called an annulus ventralis. And basically what that is a sperm pouch, a place to receive spermatophores from the male. And so what happens during mating, and that's different from our Canberrids, which do have a sperm pouch. But what happens during mating is the males will glue a bunch of spermatophores all over the ventral surface of the thorax and the abdomen. And then another different feature about signals is they typically will oviposit, lay their eggs shortly after. They attach them to their abdomen, and then they carry the eggs over winter. Whereas a lot of, especially here in Minnesota, a lot of our species will wait till spring to oviposit and then carry the eggs around. So some different life history aspects, which could be important in terms of trying to control these things. Okay, so what's so difficult about dealing with signal crayfish? And this is information from other places where they've been introduced. They're super fast growing. They can live more than 10 years. Typical, our natives, maybe 2 to 3 years max. They produce lots and lots of eggs. A single female can have up to 3 to 4 clutches during her lifetime. Like other crayfish, they're omnivores, so they can adjust their diet depending on the local environmental conditions. In certain, though, they're also highly mobile. So if conditions are moist, they can move between lakes. And then in some situations, they're known to burrow. They're not considered a burrowing crayfish, but if, let's say, water levels drop, they can burrow, which can make them more difficult to catch and control. And then another good thing for them, not so good for us, is that they can tolerate all kinds of environmental conditions. So out in California, for example, you can find them in alpine lakes all the way to floodplain rivers and estuaries. A couple good pieces of news, perhaps in terms of control, or is they take longer, the females take longer to reach sexual maturity, 2 to 4 years. Ours can be ready after just a single year. They appear to be more vulnerable to desiccation than, say, red swamp crayfish, and they know that from shipping them around. And then another idea is that, and this comes from Eric Larson, who worked a lot in the Pacific Northwest, is that because they evolved in an area which was outside of the native range of the sunfish and the bass, our favorite panfish, that they may be more vulnerable to predation by these fish, which we have here. So which could potentially be good news in terms of control. And then just generally in terms of what are the impacts? Because they're omnivores, they affect all levels of the food web. So they can shred aquatic plants, good ones and bad ones. They can predate or outcompete native invertebrates, other crayfish. They can compete directly with fish for food and habitat. And they do predate fish eggs, which could be a problem. And then if they're in certain places in Europe where they're in really large numbers, they can do so much digging in the sediments that it can affect water quality. So that's a potential impact. And then they are carriers of the crayfish plague, which has been a huge problem for European and Japanese crayfish. Here, it's probably will hopefully be less of a problem because a lot of our species are also carriers, and they have different degrees of immunity to the crayfish, to the crayfish plague, that is. And then a couple things we're really here for Minnesota we're more concerned about, or a couple things are just the potential economic effects. These guys, since they're similar to what we worry about with rusty crayfish, if they can come in and affect, they're really good at removing aquatic plants, they could affect wild rice populations. And if you're affecting aquatic plants, you may also be affecting, of course, then the game fish habitat. So both of these are economically super important and could be impacted by the presence of signals. And okay, so where were they discovered? Back in October, and the map on the right showing Minnesota, and the blue star there is right around Alexandria, Minnesota, in Douglas County. And their commercial harvester was out setting fyke nets to catch sucker minnows. And over the period of about a month, he caught 10 quite large signal crayfish, 9 males, 1 female. Luckily, the female did not appear to have reproduced, did not have eggs, but so we don't have a lot of individuals yet. In terms of potential for spread, the Lake Winona, where they were found, the red lake there, is directly connected to 2 smaller, deeper lakes. And then these are just within, a mile, half a mile, to a whole number of other lakes in the Alexandria region, including Lake Carlos, which is the headwaters of the Long Prairie River, which is, of course, a tributary of the upper Mississippi. So lots of potential there for spread to the upper Midwest, which is, of course, something we would not. Okay, so what are the plans so far in terms of how do we respond to this new invasion? Well, the goal is to try and, of course, eradicate them from Lake Winona, where they were found, and any other water bodies where we find them. And this will basically be going in 2 phases, because we don't know a whole lot yet about where they are and how many there are. The first part is going to be delineation, where we try to figure that out. And we'll use a variety of trapping and netting techniques with the idea of capturing all the different life stages, juveniles to adults, both sexes, to get an idea of numbers and where they are. And then as we're doing this, we had a great idea from the Michigan DNR, is to try and do intensive trapping between infested and we don't have this information yet, but infested and non-infested lakes, to try and confine them to where they are currently. So until the point where we can potentially think about permanent barriers. And then this summer, Gretchen Hansen's lab is going to be, and I believe, speaking with Gretchen and listening to Courtney, the optimal time for doing environmental DNA sampling would be midsummer, when the crayfish are most active. So she's going to be doing a wider spatial scale, looking at other lakes to see if signal crayfish and other invasives can be detective. And then Eric Larson is currently working on mitochondrial DNA analyses of the individuals we've captured, with the idea of trying to identify their source. Are they from Europe? Are they from the Pacific Northwest? And then using that, kind of like Courtney was talking about, to adjust the environmental DNA assay. So it's very specific for the signal crayfish population we're dealing with. Just looking at some of the part of this initial phase is just to assess, okay, which of these trapping methods or netting methods is most effective. These are just a variety of methods. I'm sure you're all familiar with the regular old minnow traps. There are also refuge traps, which can target females and juveniles a bit more. There are things called bundle traps, which are a bunch of sticks or leaves all tied together, and juveniles seem to be attracted to these things. And then our commercial harvester was quite successful with fyke nets. So we're going to use a variety of methods to see which are most effective. And then working with our fisheries folks, the idea will be to try and enhance predation of crayfish in Lake Winona or wherever we happen to find them. And that could involve a number of different management techniques, trying to protect the spawns of the important crayfish predators, potentially restricting fishing in certain lakes, but it could involve a number of things. And then so once we have all this information, the numbers, where they are, what are most effective methods, then the idea is to remove them, right? To try and eradicate them. And that will probably be a combination of really intensive trapping in areas where the signals are concentrated, perhaps some sort of game fish management to enhance predation. And then we'll also explore the idea, once we have the information, of establishing some sort of crayfish barriers, which could be physical. It could perhaps involve CO2. We're not sure yet. And then, of course, we'll be monitoring the success of the activities with more trapping, the eDNA sampling. And then more long-term, and Tina just sent us this poster, but more long-term, the idea is to do some education and outreach to the public about this. Of course, we'll continue to monitor for signal crayfish. Our group here in the DNR, Rafael, is working with organisms in trade and is doing outreach. The idea is to do outreach to businesses which use live non-native crayfish. And it is, imagine most of you know, it is legal to import live non-native crayfish for things like preparing biological specimens or aquaria or food. You just can't, of course, release them into the environment. And then just general outreach to the public. And then MACERC is going to continue their program of doing citizen science environmental DNA surveys, not only for the signals, but for zebra mussels and rusties and so forth. And this is just some of the folks you can contact besides myself about what we're doing. And I can't see the clock, Tina. How am I doing? I don't have a lot of time.
You're doing great, Don. You got
Okay.
2 more minutes if you have any final things you want to share.
Okay. No, I was going to throw in a crayfish quiz, but but I was trying to stick a poll in the chat, but I think you would have to do that for me, but that's
Okay.
fine. But this little test here is just to test the folks in the audience, see if you can pick out which 3 of these species are beneficial native crayfish, and which 3 of these are either are currently or were at one time the invasive. So you can throw in, if you'd like, throw in something in chat there, and I'll pop up the answer. But since we're a little bit out of time, these 3 guys are our most common. The virile, devil, and calico are our most common native species here. And then we've got of course, the signal. The red swamp was discovered back in 2016. As far as we know, that was the only record in the state. And then, of course, rusties are sort of all over the eastern 2/3 of Minnesota. And here, let's see.
Thanks, Don. Any questions for Don?
Got some suggestions for largemouth and smallmouth bass. Yep. And one thing I should say, talking with the fisheries folks here and in Michigan, there's some good information. For example, bluegills have to be a certain size before they're good crayfish predators. So you have to consider, when you're looking at your fish population, you have to know the age, size, structure. And that's one of the things, that's some of the data that luckily the Minnesota DNR already has. And then we'll be updating that hopefully as we go.
All right, so
And what
a new
else?
species to keep an eye out for this summer. So if you have additional ID questions, Don is your guy.
Thanks, Tina.
Yeah, and thank you, Don. All right, I am your last speaker for this session. So let me share my screen, toggle some things on and off here. And then Doug, if you want to let me know when you can see my screen and such.
Looking good.
All righty. So hopefully just going to spend 5 to 10 minutes here about a new program, or a program we're trying to do again this summer, where you can get some free stuff. So is it going to let me? Okay. So for working with bait shops and other businesses that sell live bait, wanted to provide a little background here. We know that anglers are aware, care, and take action to prevent the spread of invasive species, but sometimes they demonstrate mixed knowledge and practices. We know anglers prefer to receive, or angles prefer to receive AIS information at bait shops. Bait shop staff agree that preventing the spread is the right thing to do, and it's their responsibility to provide AIS information to customers. And local managers like yourselves are seeking opportunities to build relationships with businesses and anglers on AIS issues. So we're going to take a community-based social marketing approach to engage live bait sellers and their customers on AIS prevention, such as the proper use and disposal of live bait. So like I mentioned a little bit earlier, we did a survey in 2019 to get the behaviors, awareness, knowledge, motivators, barriers, and such for anglers to dispose of bait properly when they are done with it. And they prefer to receive information at boat launches, bait shops, and fishing accesses. A majority of them have strong attitudes towards AIS prevention. Again, we have this mixed knowledge of practices. 1/3 of them reported releasing bait, which is illegal and a risk to introduce invasive species. But they do not encounter significant barriers to proper disposal and handling of bait, and they are motivated to take action. So we developed, based on that survey, we developed some new resources to provide some clearer steps for people to follow and to increase awareness of those laws and best practices, since we're seeing kind of mixed knowledge and behaviors with those. We also want to promote social norms. For instance, the majority of Minnesota anglers take action to prevent the spread of invasive species. Gathering commitments is another big piece of behavior change. People that make a commitment are more likely to take action. And so this sticker and poster is a way to collect public commitments from anglers to properly dispose of their bait. So we did some pilot testing in 2020 and 21. So that's where we got a lot of our ideas for those new resources that I just showed. We had 2 organizations visit 6 shops, do 2 events. They created their own materials. They did some site visits and events to engage bait shop owners and customers. And they were able to gather over 100 commitments in just that small project window. And then what we did is we learned from those small pilots and created kind of a statewide pilot in 2023 called Engaging Bait Shops. And we had 6 organizations visit 17 shops. We had the free materials that I showed provided by us. And we did some store visits to engage bait shop staff and have them distribute materials to their customers. And by we, I mean the participants did this. And each visit only took 15 to 20 minutes. They visited each store 3 times. So it's a very small time commitment to get this information out to bait shops. We also did some pre and post program surveys to track change in awareness and attitudes. So for the 23, what did we learn? For the barriers and motivators for providing AIS information to customers, what makes it difficult is time, knowledge. The customers are not interested or the employees are not interested. And then what makes it easy is the materials that we provided, which was great to hear. All the participating shops agreed to continue to hand out those materials. We sharing knowledge during the visits between the local AIS managers, the pilot participants, sharing information with the bait shops was something that they promoted. The printout from the license sales, that acknowledgement that's on non-resident fishing licenses was helpful, and just talking boating and fishing. So you can see that the materials helped them better understand customer questions about invasive species, and that they agree that the business has a responsibility to provide AIS information. So the shops want to provide this information. It makes it easy for them to provide AIS information. So what's next for 24? We want to recruit more participants. So let's see if this works. Yes, there is a form in the chat if you would like to participate in the outreach efforts this year. It's an opportunity to build relationships, educate businesses and customers, and evaluate outreach efforts. Fill out the form that I just linked in the chat, and we'll be in touch. You get some free stuff out of it too. So some of the things we're changing for 24 are simplifying the forms and the evaluation piece, sharing experiences. We're going to have a kickoff meeting this spring with some past pilot participants to share their tips and tricks. And we're also going to get the materials out earlier this year. Last year was a little bit late into the fishing season, so we'll plan to get the materials out even in mid-March so you can start reaching out to those bait shops. We also want to expand the opportunities, so not just bait shops, formal bait shops, but other businesses that sell live bait like resorts and outfitters. And we also want to expand it to events such as Kid Fishing Days, which would be a great location to implement some of these things, tournaments, booth stores, other options. We're open to other ideas as well. Ways that we can engage anglers that use live bait to properly handle and dispose of their bait after they're done using it. So a few little last things on evaluation. It's a really important piece of any behavior change approach. And through this kind of pilot 2.0 is what I'm calling it, working with businesses that sell live bait and events and angling events, we want to collect this kind of qualitative and quantitative data on how much time it takes to reach out to this, what additional support is needed, are shops interested, what are the awareness levels, what is the change in awareness levels and behaviors, all sorts of things about how we can really better engage and build relationships with bait shops and other places that sell live bait to promote proper handling and disposal of live bait to prevent the spread of aquatic invasive species. So, any quick questions on that?
Just like to remind everybody that there are some comments, resources, and also a question for Tina in the chat from Tim Terrell. And he asked, is there a metric to show that the percentage of people return their bait or dispose of it properly when they receive the information from the bait shop?
So we aren't interviewing through this project in particular. We aren't interviewing the customers. We're interviewing the bait shop owners and employees. So in the future, we would have to interview anglers themselves about that. So right now, it's just the shop owners. Any other quick questions on that?
Not seeing anything in the chat.
All right. Well, that kind of brings us near the end of our workshop. I want to ask, did anyone have other questions that came up for any of our speakers here this morning? Then I'll ask another question. Are there any, Charlie, go ahead.
Hi, I don't really even know if this is much of a question, but the crayfish from before, I was really wondering about tying the eDNA, to that research, and more or less just interested in an opinion as to, that they were discovered in that one particular water body, but yeah, I wonder, it just makes me wonder how long they've been, misidentified, how long they've been there and such. So I don't really, I guess I don't really know. Is like anybody have a prediction as far as, how widespread they are, or did somebody actually catch them early?
Yeah, no, great question. And short answer, no, we have no idea. This is the only record that we know of. And this was just to give you an idea, the commercial harvester that found them, he regularly does this lake. He nets in this lake, let's say, twice a year. However, because of COVID and some other things, it had been about a year and a half since he'd done some intensive trapping in that lake. And so when they were introduced is pretty hard, but there's sort of like a year and a half window of when they were potentially introduced into that lake. And we had a brief period of time to do some follow-up trapping in some of the connected lakes. And we did not, with regular minnow traps, we did not catch any signal crayfish. We caught a bunch of the natives, so we know our traps were working, but we did not catch any signals. But like you're saying, we don't know. They could be released somewhere else. And if anyone sees any crayfish, of course, with big red underclaws, please contact us because, it's just, and then in terms of the environmental DNA part of your question, that was, I think, the whole motivation for Gretchen Hansen and her lab to get more citizen scientists involved in surveying, getting out there with these kits, and so we can get some sort of detection all over the state, and I'm not sure how widely it's distributed. But it does depend on, and Courtney can talk a lot more about this, but it does depend on the time of year you sample. Like if you do too early and the crayfish are just sitting there, you're not going to detect eDNA. Middle of summer when they're being super active and eating and excreting is like optimal time from what I understand.
Thanks, Don.
Thank
Kyle, go ahead.
Yeah, 2 questions, if that's okay.
Mm-hmm.
One's for Tina about the pledge. And as an AIS coordinator, do you know if it would be legal to offer an incentive for folks to take the pledge, say put their name into a monthly raffle, that kind of thing?
As a state agency, we can't do that.
Okay.
But you could potentially look into your county ordinances and see how that might work for you. We do not collect contact information through the pledge. We only collect names because we don't want to have that data. We just want people to pledge to protect Minnesota waters.
Sure.
But you and I can talk more about how that might be more applicable, and how I'd love to know how other people are using the pledge and getting that out on the landscape. And we intend to, keep that moving this summer as well. So.
Sure. Thanks. Yeah, if we could talk about that later, that'd be great. And then a quick question on the eDNA. Maybe this was kind of answered through Doug's comment on the positive results, but I was wondering if anyone's kind of done some research on how far of an area that DNA can spread out of where that organism would be, if that makes sense. So essentially, like in an area without much flow, if there's a zebra mussel in one bay, is there any kind of information on how far that is shown?
That's a really good question, and that's an active area of research. If there are connected lakes, they've shown that the DNA from species that are up in upstream lakes can flow into downstream lakes where there actually isn't a population of a certain species of fish there. And so it can go, a matter of meters. I wouldn't say that it goes miles and miles, but who knows? Like once we get more and more samples out there, we know it can persist in an environment for days. And, as Doug said, like 7 days for carp DNA in the Chicago River. So this is an active area of research. But that said, it does degrade over time, and you're getting small amounts that travel those distances. If you get large amounts of DNA, it's probably pretty close to a source.
Sure. And then those tests, do they show kind of the amount of DNA in a sample?
Yep, they do. So it's called quantitative PCR because we are able to put a quantitative number associated with it.
All right. Thanks, Courtney.
Yeah, thanks, Kyle. Thanks, Courtney. John, you want to take our last question for the workshop here?
Yes, I had, thanks. I had put it in and Don had answered this. I had asked if that contaminated lake had been quarantined, if there's an immediate response or rapid response to finding a new AIS. Is there anything in state law, regulation, DNR policy that quarantines the lake? Or is there any attempt to do that now?
I'll ask what your thoughts are on quarantine mean, because it depends on what you mean by that.
So no one goes in or out of the lake unless they have been fully decontaminated.
So Don, if you have a response.
Yeah, the only thing is that would refer more to the prohibited species like zebra mussels and so forth. And right now, because signal crayfish is brand new, it's what's the term, Tina? Is it unlisted?
Mm-hmm. Unlisted non-native.
Unlisted non-native species. Mm-hmm. So there's not a specific regulation, dealing with signal crayfish at the moment. Mm-hmm. But obviously, the rapid response part would be, as I mentioned, as soon as water temperatures warm up and the crayfish are more active, the idea is first to figure out if they're still there, go in with a bunch of trapping, but then at the lake connections to do intensive trapping. And there is, in Lake Winona, there is a carp barrier already because there are invasive carp there. There are not zebra mussels or rusty crayfish, but a carp barrier would not really hold signal crayfish, so especially the young ones. So, but the idea is to do so much trapping at the lake connection, like to hire a local harvester to do that there'd be minimize the chance that a crayfish could get from one lake to the next directly.
Thanks, Don.
That's the current plan, and then eventually trying to get something permanent established, but of course based on what we find, which we don't know yet.
Thanks, Don. All right, we've got 2 minutes left. I'm going to have one more ask for everyone. If you are willing and able, turn your camera on so that we can see everyone's faces that was here today. We had over 60 people participating, and I always like to take a little screenshot of all the people that attend. So for those of you that can, turn on your cameras, and we'll take a little screenshot of everyone. So say, protect Minnesota waters. So, all right, 1, 2, 3. All right. Thanks everyone for participating in that. And I think with that, we'll say it's 11:59. We are wrapping up our final workshop. Thank you everyone for coming and participating today. Thank you especially to our speakers for preparing your presentations and sharing all of this awesome new approaches to aquatic invasive species and management. We've got 3 more online workshops, 2 more in-person workshops. So if your brain isn't already full, we've got more coming down your way for the next month and a half or so. So thank you again, everyone, for coming, and have a lovely day.